For this introduction. Thank you very much. I'm going to share my screen. So you see the slides and not the presenter mode anymore, right? Yeah, perfect.
Yes.
Great. Well, thanks a lot. I'm going to start by a quick plug because this is the online seminar, but we are having our conference, the MOB conference, in less than 2 months, or a bit more than 2 months, sorry. And this time it's organized on Heron Island, beautiful Heron Island in Australia. We've extended the registration until the end of this week. So for the few of you that have not registered, please do so in the coming days. We have an exciting program of invited speakers and that are coming from all over the world, including Australia and Asia. This is actually the real room where the conference is going to happen, overlooking the reef. So it's a pretty nice location. And for the first time in MOB history, we also have a workshop the following week, which Marco is co-organizing. And during this workshop that will allow those who want to make the most out of their trip to Australia, we're going to propose some thematic sessions. So there will be a day on science education, a day on numerical modeling, a day on microfluidic and cell tracking, a day on PIV, one on microsensor, and I will finish with a day on chemical extraction and in situ chemotaxis assay. For each of these days, there will be a theoretical part as well as a practical part. So I'm looking forward to it. I'm sure it's going to be a lot of fun. And as I said, for those who have not registered, please do so in the coming days. I'll, of course, be happy to answer any question about the conference at the end of this talk. But for now, I'm going to, as Marco said, talk about some of the work that we've been doing recently about complex chemically mediated microbial behavior in the ocean. So most of my work focuses on chemically mediated interactions occurring in the pelagic ocean. And although this environment seems very homogeneous for us, it is in fact highly structured at the scale of microorganisms. This is how microbes see the ocean as a succession of chemical hotspots coming from various sources. Some of these hotspots can come from sinking particles, from excretion or lysis events, and some can come from the slow exudation of photosynthase from phytoplankton cells. Motile microorganisms can exploit this patchy seascape by using a behavior that's called chemotaxis. Chemotaxis is the ability of microbial cells to direct their movement in response to a chemical gradient, here coming from a phytoplankton cell. And this behavior allows cells to home in on nutrient concentration that are orders of magnitude higher than in the bulk seawater. It also allows cells to come in proximity to one another and to potentially start interacting. Chemotaxis has been studied quantitatively for the past 60 years. The first method to quantify chemotaxis was the capillary assay. Very simple, a capillary tube filled with a chemoattractant is plunged into a bacterial suspension. The chemical diffuses out of the capillary and microorganisms can respond by swimming into the capillary where they can be quantified. Since then, a range of different techniques have been developed to quantify chemotaxis, and the latest kind are microfluidics, which enable to establish carefully controlled gradients and to track the behavior of cells through time. However, all these techniques share one common limitation. They are all restricted to laboratory use, which means that we cannot take them into the field to quantify chemotaxis. So our idea was to use the general principle of the capillary assay, but combine it with modern microfabrication techniques used in microfluidics to finally take chemotaxis assay from the lab to the ocean. Our platform is called the ISCA for in situ chemotaxis assay. It has the size of a credit card and is made of an array of wells. Each well can be filled with a different substance, and upon deployment in a marine environment, this substance, this chemical, diffuse out of the wells, creating concentration gradients that are analogous to the hotspots I was telling you about a few minutes ago. Motile and chemotactic cells can respond to this gradient by swimming into the wells, and that's where we can sample them after retrieving the device from the environment. Of course, this is a very sanitized view of how the ISCA work, and it took us several years to get to this point. That's where we wanted to be, but it took us several years to get there. And during that time, I was working with then PhD student Ben Lambert from the lab of Roman Stoker. And with Ben, we tried, we tested a range of well geometry, of materials, and after numerous trials and error, we ended up settling on the final well design, which is what we still use today. So the ISCA wells are made of a reservoir that's 110 microliters, linked to the outside environment by a single port of a diameter of 800 micron. The first thing that Ben did when we settled on this final design was to run numerical simulation to characterize the diffusion of chemicals in this well geometry. And what he established is that when we load chemicals in the wells at a 1 millimolar concentration, it will take approximately 24 hours for the concentration to equilibrate between the inside and the outside, which means that we will be diffusing chemicals from this well for an extended amount of time. The chemical plume will also extend for a certain distance based on diffusion. And then quantify that it will extend at least 2 millimeters before it's no longer sensed by most bacteria. So I'm going to quickly run you through, explain some of the tests that we initially did to test this device. We used a smaller footprint of the ISCA that we embedded on a microscope slide into this microcosm. And we filled this microcosm with a model bacterium, Vibrio coriolyticus. With this setup, we were able to visualize bacteria as they were entering the wells. This is the kind of videos that we were able to record within the ISCA wells, comparing wells containing a chemoattractant, here mine broth, with wells containing only filtered seawater and acting as controls. Of course, we wanted to be a lot more quantitative than just recording videos. So what we did was setting up experiments where we imaged a single frame within each well from the bottom to the top every minute for 60 minutes. So that's the kind of data that we got going from the bottom to the top. Each dot on these images represents a single bacterial cell. And with image analysis, we were then able to quantify the bacteria on each frame to generate this type of plot, where you have the time in minutes here, so from 0 to 60, and here that's the bottom of the well to the top of the well. So bacteria will enter from the top of the well, and what you can see is that in the well containing a chemoattractant, here mine broth, you have bacteria entering after only a few minutes, and after 40 minutes, between 40 and 50 minutes, bacteria are homogeneously distributed in the entire depth of the well. In comparison, that's what the control wells look like with no accumulation at all. Another way to look at this data is to integrate the number of bacteria on each frame at every time point to build this accumulation plot. Where here you have the number of bacteria entering via random motility in the filtered seawater wells, the controls, and here the number of bacteria entering in mine broth wells. Of course, that was how we tested the ISCA initially, quantifying bacteria with the microscope at every minute. But when we deploy the ISCA in the environment, that's not what we do. We deploy the device for 60 minutes, we retrieve it, and then we count the number of cells in the wells. So we're only looking at what's happening in a snapshot of time after 60 minutes. And if we take this data and plot it as if it was the data I'm going to show you later, so that's just an example. This is our two treatments, so filtered seawater and mine broth. And on this y-axis, we have what we call the chemotactic index, which is the number of cells in a given treatment, here mine broth, divided by the number of cells in the control wells, in the wells containing filtered seawater only. So it means that the chemotactic index for the filtered seawater control is always equal to 1. If your chemotactic index is significantly higher than 1, as it is here, you have an attraction. And if it's significantly lower than 1, it means that you have a repulsion. Of course, I don't need to pitch that to this crowd, but this theory it works well in the lab. This diffusion works very well, but as you all know, the natural environment is much more dynamic than the lab. And the nice diffusion from the well that I was telling you about that allows the migration of cells inside the ISCA well will be constrained by any flow that will run in the field, washing away this chemical plume and preventing any chemical gradient to establish between the inside and the outside. So to be able to detect chemotaxis in the ocean, what we had to do was to suppress the influence of local fluid flow by enclosing the device into this flow damping enclosure. So they are transparent. We put one ISCA per box, secure it, slowly fill it in situ, and then we completely seal the box. So the box is closed from the outside environment, and the fluid flow is therefore drastically reduced. As you can see from this picture, this process can be done in parallel and allowing multiple ISCAs to be deployed at the same time in the same body of water. I told you that we can count the number of cells after retrieving the ISCA, but that's not all we can do. We can also determine who they are and what they do by sequencing their DNA. And we can finally isolate them to further characterize their chemotaxis capability in the lab. We've mostly used the ISCA over the past couple of years to characterize phytoplankton-bacteria interactions. And that's because interactions between phytoplankton and bacteria are at the base of the marine food web, and they are central to the structure of marine ecosystems. So using this approach, using the ISCA, we've started working on who interact with whom, elucidating what kind of bacteria and archaea are attracted to what type of phytoplankton. We've recently started working on the effect of stress on phytoplankton, how this affects the chemical they release, and how in turn this affects chemotaxis. And that's, we have a couple of manuscripts in preparation. And most recently, we've started to look into the kind of molecules that are the strongest chemotactic signal in situ, trying to investigate what really attracts bacteria in the field. This is the latest work that we've been doing, and that's what my talk is going to focus on today. To understand what kind of molecule attracts prokaryotes, one of my postdocs, Maeva, collated a database of nearly 700 molecules that have been tested in chemotaxis assay over the past 60 years. So those are all the molecules that have been tested since the beginning of chemotaxis assays on prokaryotes. There's a wide range of different molecule classes, but looking at the identity of the chemical tested was quite revealing because 85% of the molecule tested have a low molecular weight. They are on this side of the graph. And the reason why small molecules have been more tested than others is because they are assumed to be better chemotactic cues because they diffuse faster. However, we know that many organisms also release much larger molecules that will be on this side of the graphs. Do they also attract or affect the behavior of microorganisms? We don't know because most of these molecules have not been tested. So one of my former PhD students that I co-supervised with Roman Stoker, Estelle, tried to address this question by looking at the size range of molecules. Estelle used the ISCA to test the chemotaxis towards mannitol and glucose, which are two small molecules, laminarin bios, which is a dimer of glucose, and laminarin, which is a polymer of glucose with side chains of mannitol. So as you can see, All these 4 chemicals are linked. And importantly, laminarin accounts for 25% of the annual carbon pool derived from primary production in the marine environment. It's a very important storage compound for brown algae, and it has relatives, so molecules of the same structure in many phytoplankton. So in a nutshell, it's like asking if bacteria prefer to chemotax towards small pieces of chocolate, larger piece of chocolate, or the entire chocolate bar. To test this question, Estelle and I went to Bergen in Norway, where we took part of mesocosm experiment that was run by the lab of Asaf Vardi. At the start of this experiment, these mesocosm bags were filled with fjord water and were enriched with nitrogen and phosphate with the hope to initiate a phytoplankton bloom. We were one of many teams taking measurements in this bag, and fortunately for us, we saw a phytoplankton bloom. I learned when I was over there that it's not always a given, so we were quite lucky. We saw a first bloom of the picophytoplankton Micromonas, which peaked after day 9 and collapsed, followed by an even larger bloom of the Coccolithophore Emiliana exlei that peaked and collapsed. With Estelle, we deployed ISCA throughout this bloom, and this is the results for laminarin. So that's the largest molecule, the large piece of chocolate. The thing I want to point out here is that when the bar at a given time point is empty, it means that there's no difference between the number of cells in laminarin versus the control, filtered seawater. And when the bar full, it means that there is significantly more cells on these time points compared to the control, okay? So what you can take away from this graph is that the large molecule laminarin adds a strong but viable induced a strong but viable chemotactic response. At the very end of the bloom, we had a chemotactic index of nearly 6, which means that we had 6 times more cells in the wells containing laminarin compared to the control. That was surprising to us. It was very nice to see this large molecule being a strong chemotactic cue. But what was even more surprising was that none of the small molecule induced any significant chemotaxis throughout the bloom. And these includes molecules such as glucose that have been used extensively in laboratory in lab-based chemotaxis assay. So we decided to focus on laminarin, and we sequenced the bacterial communities that responded to laminarin at each of these time points. As you can see, the community in the laminarin wells, that's the ISCA wells containing laminarin, were very different from the seawater surrounding it. And Estelle was able to identify bacteria that were significantly enriched in these laminarin wells. Importantly, what we did as well was to isolate bacteria from these wells. And Estelle was able to isolate 4 bacteria that had 100 DNA sequence identity at the 16S level, with those that were enriched via DNA sequencing. So those identified by DNA, we were able to isolate 4 of them. So park this information because we're going to use these isolates in 2 slides. Okay. So I told you that chemotaxis to laminarin was strong but viable. And of course, we wanted to understand the cause of this viability. So what we did was to use the large amount of data that was recorded daily by the other teams that took part of this large experiment. There were 61 variables in total that were measured daily on these bags, and that included concentration of monosaccharides in the water, concentration of glycans, which are larger molecules, a range of inorganic and organic molecules, some biological variables such as the amount of certain class of eukaryotes, bacteria, and viruses, as well as physical variables such as salinity, temperature, turbidity, etc. So what Estelle did, she correlated the chemotaxis to laminarin with each of these variables, and she identified that the strongest correlation happened with the concentration of DMSP in the water. DMSP stands for dimethylsulfoniopropionate. So DMSP is a ubiquitous sulfur compound that's produced mainly by phytoplankton, but also by certain type of bacteria. And it's as I said, ubiquitous in seawater. You can take a seawater sample anywhere in the ocean, you will be able to measure this compound. So what this result means is that when chemotaxis to laminarin was high, concentration to DMSP in the water was also high. Of course, for the student in the audience, correlation does not equal causation. This is real data coming from the US, where you see this near perfect correlation between the consumption of mozzarella cheese over the past 10 years and the number of PhD awarded in civil engineering. So it's always good, even when your correlation is perfect, to double check it, because it doesn't mean much. And that's exactly what Estelle did. She took advantage of the fact that we had these 4 isolates from the Iska wells enriched from laminarin, and she was able to set up experiments to test this hypothesis. So she filled Iska wells with laminarin, as she did in the field, and she spiked the surrounding seawater with various concentration of DMSP. Importantly, DMSP was not in the well, it was in the water surrounding the Iska, like in Bergen, like in the field. What we saw was that when for the 4 isolates here, when there was no DMSP in the water, that's the bar in gray, we had strong chemotaxis towards laminarin, but when DMSP was present, the chemotaxis towards laminarin was stronger. This effect didn't always happen at the same concentration. Sometimes it happened at high concentration, for others it happened more at lower concentration, but it was always present. So of course, we were very pleased to confirm this link between the 2 molecules, but we wanted to understand what was causing it. The first thing we thought was that DMSP might be increasing the swimming speed of the bacteria. It's been reported for other strains before, and that's exactly what we tested. But unfortunately, with or without DMSP, the swimming speed of the bacteria didn't change. And so were a range of other swimming parameters that we tested. We looked for percentage of motile cells, turning frequencies. None of these swimming parameters were really affected by DMSP. So we digged a bit deeper and identified an analog of our behavior in a classic paper from 1967. In this paper, Adler and colleagues noticed that E. coli was more attracted towards serine when methionine was present in the water. This was concentration dependent, so the more methionine was present in the water, the more chemotactic the bacteria were towards serine. They even went one step further and identified the mechanism behind this, as methionine was acting as a methyl donor, donating a methyl group to the chemotaxis sensing apparatus of the bacteria, increasing the gradient detection capability. Now, DMSP is called dimethylsulfoniopropionate because it has 2 methyl groups. So we were wondering if similar processes could happen with DMSP. Can DMSP donate one of its methyl group to methylate the chemotactic sensing apparatus? We tested this hypothesis by replacing DMSP in the water with methionine, and we saw a similar increase. When methionine was present, chemotaxis towards laminarin was increased. We substituted methionine for choline, which contained 3 methyl groups, same effect. And then we tested the effect of homocysteine, which is a molecule that is very close in structure to methionine, but does not contain a methyl group. And there we saw no effect at all. When homocysteine was present, there was no difference in the chemotaxis towards laminarin. We did a range of other tests to confirm this, the fact that DMSP was providing a methyl group. And the outcome was that this interaction between DMSP and laminarin is, to our knowledge, the first example of 2 molecules that synergistically affect the behavior of marine bacteria. Taking a step back and going back to the bigger picture I was telling you about at the beginning of this talk, none of these hotspots that I was telling you about release only one molecule. In fact, they most likely release hundreds to thousands of molecules. If we look at the diffusion profile of different molecules coming from these hotspots, so imagine here at 0, we have a phytoplankton cell. And we're looking at the concentration profile of chemical that will diffuse away from this phytoplankton cell based on different chemical classes. We have here some diffusible compounds that small molecules that will diffuse fast and create weak gradients. So they are, if you want, long-range signals. And at the opposite end of the spectrum, we'll have much larger molecules like laminarin, which will create much sharper gradients and will diffuse less far away from the cells. What is happening where these gradients overlap in terms of chemotactic detection and synergistic effect, we simply don't know. So to start looking into these processes, one of my current PhD students, Bhuvan, started developing a new chemotactic platform that's called the chemotaxis choice experiment, or ChemChoice. It's a lab-based experiment. It's not something that we can take to the field yet, but its novelty is that we can seed 4 different chemicals or 4 different chemical mixtures. They don't have to be pure chemicals, but 4 different mixture or pure chemicals that will be inserted and then mixed through the channels and come back through this outlet here. The flow is constant, so it's a steady gradient. We don't stop the flow, so we wait for the gradient to be formed. We keep a very low flow rate, and then once we're ready, we inject bacteria from the outlet. The bacteria are able to swim against the flow, and at each intersection make chemical decision, what we call chemical decision. So they're basically able to turn left or right, depending on what chemical they prefer. We've just started to use this device, so I'm going to show you some data that we gathered in the past couple of months. And the first thing we wanted to do to test this device was to input 4 chemicals that are strong chemoattractants to see how bacteria would choose between them. So the first thing we did was to take a step back and do an ISCA experiment with our model bacterium Vibrio coriolyticus. So we tested 6 different chemicals in isolation. Okay, we tested 2 sugars and 4 amino acids. And 5 of them elicited a strong chemotactic response. That's the chemotactic index here. So we selected these 5 chemicals for further experiments with ChemChoice. That's the architecture of ChemChoice, as I was telling you. So in the first experiment, we had galactose, lysine, alanine, and histidine. That's the results of the chemicals in isolation from the ISCA. And the first thing we did was to look at what happened at the main intersection. So what was the first choice that these bacteria were making? 100% of the cells went for alanine and histidine. So they turned down instead of up at this intersection. There is no error bar here because despite the fact that we repeated this experiment 3 times, there was never a single cell that went up. So the behavior was really striking. 100% of the cells were going towards alanine and histidine. But what happened downstream was even more striking because 99% of the cells went for alanine only. Now you might think, okay, but that's kind of predictable, right? Because alanine is the strongest chemotactic signal in isolation. That's the molecule that elicits the strongest chemotactic response. All right, let's swap alanine for serine. Serine has elicited a chemotactic index that's similar to histidine and lysine, and let's see what happens. Similarly to alanine, 100% of the cells went down towards serine and histidine. And then 99% of the cells went for serine only. We were thinking, okay, this might be a case of chemical interaction. It might be that when an amino acid is next to histidine, it will attract cells more. So we swapped all the positions possible, but the results remain. When Vibrio qualiticus are exposed to these 4 different chemical signals, they only go for one. So as a take home from this talk, I showed you 2 things today. When 2 abundant molecules, DMSP and laminarin, are together in the ocean, their effect is synergistic on chemotaxis. As the opposite, when we take these 4 smaller molecules, bacteria tend to go for only one of them and disregard the rest. So I hope that I convinced you that we are only looking at the tip of the iceberg when it comes to chemical interactions and their effect on behavior. And fortunately for us, we're now at the point where we have the technology to start probing these synergies and better understand the effect of chemical mixtures on microorganisms. And that's what we are aiming to do in the coming year or coming years. So with this, I would like to thank all the people that have been involved in the work I've presented today, and I'm happy to take any questions. I realized that I was a bit quick. It's 30 minutes only, but that will give us a bit more time to discuss if people want.
It's perfect. Thank you, JB, for this very engaging talk. Marco, do you want to start?
There was also, I think, Luke put
oh, I'm sorry. Yes, there was the question in the chat, and the question reads, Luke, do you want to ask the question now?
Yes, can you hear me?
Yes.
Yeah.
Yeah, I was just wondering, like, if you could do this chemotactic assay, like, why don't we visualize the thing in situ? I suspect that it's much more complicated, but I was just wondering if anyone had done that before.
No, although there are some microscopes that you can now take in situ, we've never done this. I know that other people were attempting to do this, but I do not know if they succeeded.
Okay, thanks.
Marco, go ahead.
Okay, so about the second part of your talk on the chem choice.
Yeah.
Did I understand correctly that you swap the 4 positions around?
Yeah.
Okay. So have you tried to do sort of compensatory concentrations to try to convince them to go one way versus the other way, just by upping the concentration of one versus the other 3?
No, we have not.
And then try to look at the choice when you are in a situation where you balance, let's say, one with the other. Meaning, in your assay, right? You have that the different molecules will also have different affinities. For example, for the receptors, I guess, that are on the surface of the bacteria, and therefore, if I have the same concentration of 2 things, one of which has a much higher affinity, then perhaps that will mask whatever is the signal from the other one. So maybe I should sort of balance by putting more of the other one. I don't know if that makes sense.
Yeah, it does. It's a good point, but we have not tested this. We literally got some of this data a few months ago, and we're trying to take it to the next level now by looking at mixtures, but we have not modified the concentration. So everything that I showed you and everything that we've tested has been at the same concentration.
And JB, did you said that you measured the ICs on these individual compounds in ISCA before you put them into this machine for decision making?
We wanted to know, without any competition between the molecules, what will be the index, so the strength of the chemotaxis towards individual compounds.
And do you have ISCA measurements for the mixture of all these all of these compounds as well?
I'm not sure how I would get this. So you would mix the 4 chemicals in the ISCA well?
Yes.
No, I do not have that. No.
Okay. It's very interesting, the ChemChoice results. Arnold?
Can I ask a question? Yeah, really interesting results. Yeah, my question is just about the timescales. You know, how long does it take? You know, in these smaller devices, I think it's probably much faster than the previous experiments where they have to swim through a whole well, right?
You mean the how long does it take for a ChemChoice experiment to take place?
Yeah, exactly.
About 25 minutes when we start injecting the bacteria. Yeah.
Okay. Yeah.
But then counterintuitively, even though the experiment is faster, the data analysis is much longer because we're dealing with cell trajectories instead of counting cells via flow cytometry. So if you ask me what's the quickest to get from experiments to results, the ISCA is much quicker.
Do you know if like something changes in like the upregulation of the chemical receptors? Is there enough time for that or not really?
I do not know. I do not know, unfortunately.
Okay. Nice.
Johannes?
Hey, JB. Thanks for this interesting talk. I was wondering about this this ChemChoice experiment. Did you try or consider basically flooding the entire device with one chemoattractant? So, for example, if you would basically have serine everywhere, would it still be able to find the histidine concentration? Because I think it's going to be very critically depending on which chemoreceptor is sensing what, and in what ratio are these chemoreceptors actually expressed within the chemosensory array. And one way to find that out without genetics might be just to see to what extent these signals are additive or not.
That's a very good point. What we did that I haven't shown you was testing single chemicals and moving them across to make sure that the ISCA results were reproducible and that each chemical in isolation, if we had let's say galactose and 3 branches of seawater, would we see still a chemotactic response in the ChemChoice? And we did. For all the chemicals we tested, when we test them in isolation, we see chemotaxis. But we have not flooded the device with a single compound and tried to superimpose gradients. It's a good point as well.
Are there more questions? I have another question. Go ahead.
Sorry.
Hey, how are you doing?
David, good to see you. Yeah, I have a question. I'm taking you back to the DMSP story at the beginning, because I had some thoughts there. And we know that one of the interesting effects with chemotaxis when you go to the open water is that you don't actually get much. And if you take a liter of water and then you put it overnight in the lab, you get much more chemotaxis the next day.
Yeah.
Right? And we often think of that as maybe the effect of all kinds of plankton and phytoplankton that are dying and releasing stuff and enriching the water overnight.
Yeah.
So is it possible that I love the hypothesis with the DMSP and enhancing the chemotactic ability, but maybe it's also just that when you have a higher concentration of DMSP, it actually means a more enriched parcel of water, which is already enhancing or eliciting more chemotactic movement.
Yeah, absolutely, Ho. It's totally possible. But what's likely in the case of DMSP and facilitating the chemotaxis towards larger molecule is that, as you know, DMSP is a small molecule, so it diffuses much faster. And so, as I said during my talk, if you take a water sample anywhere, even in the open ocean, you will still find millimolar concentration of DMSP. It's always there. So you can assume that when we're far away from a patch of phytoplankton, you will have this homogeneous background of DMSP at low concentration that can still act as a methyl donor and can still facilitate the gradient sensing capabilities towards sharper gradients, stronger gradients around cells, such as large molecule like laminarin. So I think that's the difference in diffusion between small molecules like DMSP and larger molecules that enhance and really makes this synergy works, if that makes sense.
Yeah, I think so. Okay.
Hey, Oscar.
Hi, can you hear me?
Very good.
Yes. Yeah, I was wondering about these chemotaxis towards large molecules. It seems to me that this would make a lot of sense in symbiotic interactions or more stable interactions between phytoplankton, for example, and bacteria. So basically because the signal is more localized, slower to diffuse away, and it's also more expensive to produce. So it makes sense that you want to invest in something, a long-term relationship. So I was wondering if you have any evidence that could support this. I don't know if the kinds of bacteria that perform these chemotaxis towards large molecules are also more localized into phycospheres or have
no, unfortunately, I don't have hidden data that I didn't show you. No, that's all I got. It's possible, it's a good hypothesis, but I don't have anything to substantiate this beyond what I showed you today.
Thank you.
Thanks. François?
Hello, Jean-Baptiste. Very nice talk. I had some curiosity questions on the chemo choice. First, out of curiosity, how many bacteria do you count when you get this 100% bar? Like, are we talking 100, 200?
It's more in the 1,000 range. Yes.
1,000 range, and you have one, not one cross the other way. Okay, that's very impressive. And then the other thing I was wondering is how do the dimensions of your channel and of your turns compare with the mean straight path of these bacteria? If it was tuned in any way to their navigation.
Yeah. So I think the mean run length of Vibrio corioticus is around 50 micron. I think if somebody knows, correct me, please. I think it's around 50 micron. So the dimensions are much larger than this. The diameter of the main outlet is 600 micrometer.
Okay.
And it reduces every time you go up.
Yeah, so 300 and 150.
We compensated the width to accommodate for no difference in flow rate. Otherwise, if you get the same width, you increase the flow rate.
Yeah. Okay. Thanks for this info. Thanks.
Any other question or any questions regarding the upcoming conference? Don't hesitate.
Maybe I have a final question. In terms of field measurements, you showed these results of chemotaxis to laminarin as a function of time for the 2 blooms in Norway.
Yeah.
Is it state of the art in terms of time resolution of how chemotactic response changes? I wonder what's the relevant time scale, if we could even partition it and measure it every, I don't know, 1 hour. It's just wishful thinking, but I wonder how quickly these landscapes of response change. Do we know?
It's a good question. I don't have this answer, but we never had the manpower to do this. perhaps this is something that's going to change on Aran Island. I might be able to convince people to do an hourly measurement of chemotaxis on the reef. Who knows? We'll see.
Well, thank you for doing it daily. Okay, I think we can stop here. Thank you for your talk, JB, and for all the interesting questions. Let's give another round of applause for JB.
Thanks.
And thank you, everybody. I think we have one more talk before the conference in Australia. Next month, we'll have Gita Jesus from UCSD. And have a good day.
Bye.
Bye-bye.